Hello class, welcome to my current events presentation on the gut microbiota involved in leptospiral infection. So, some background information is that leptospirosis is a zoonotic bacterial disease, and zoonotic means that it can be transferred from animals to humans or from humans to animals. The main animals that harbor this bacteria are cows, pigs, horses, dogs, rodents, and various wildlife such as raccoons and deer. Some modes of transmission are contact with infected bodily fluids, such as urine, not saliva, bacteria to skin contact, bacteria to mucous membrane contact, and contact with or ingestion of contaminated water. Now, as far as bodily fluid transmission goes, urine is the predominant bodily fluid responsible for diffusion of this bacteria. So if an animal that is harboring this bacteria urinates somewhere, then the bacteria will stay there until picked up by a new host. As far as symptoms go, they differ between humans and animals. In most cases, animals don't show symptoms like humans. Their job more so is to harbor and diffuse the bacteria, while human symptoms include high fever, abdominal pain, diarrhea, vomiting, and organ failure. Now, the three main organs affected are the kidneys, liver, and lungs. The significance of gut microbiota play a role in immunity and digestion. So they fight off disease, infection, and viruses, and also play a role in nutrient acquisition, which I will further discuss in the next slide. The microorganisms discussed in this research were leptospira interrogans, firmicutes, and bacteroidetes. Leptospira interrogans was the specific strain of leptospirosis that was injected into our test subject that I will mention later. Firmicutes and bacteroidetes are specific bacteria found in the gut microbiome. Leptospira interrogans is a gram-negative, sphere-shaped pathogenic bacteria. Firmicutes are a endospore-forming gram-positive bacteria responsible for digestion in the form of fermentation. Now, the fermentation that firmicutes participate in break down carbs and starches that residential enzymes in the mammal gut microbiome would have trouble digesting. As far as the endospores go that are being reproduced, they are extremophiles, which means they can survive in extreme heat or extreme cold, and they are resistant to desiccation and UV. Bacteroidetes are a non-motile flagellated gram-negative bacteria mainly found in the gut microbiome of the mammal. They play a role in digestion in the form of fermentation and immunity. So the fermentation that bacteroidetes participate in is glucose digestion, mainly polysaccharides. The product of this glucose fermentation is short chain fatty acids, which in turn gives the host energy. Bacteroidetes play a part in immunity because they trigger a T cell mediated response to kill pathogenic bacteria. So T cells are cells that develop from stem cells in bone marrow, and they target specific foreign substances such as leptospirosis. This is what they look like. The methods of this research include three groups of mice. So one group is a control, so they are not treated or injected with anything. One group is treated with antibiotics and injected with leptospira interrogans. And the last group is treated with antibiotics, injected with leptospira interrogans, and treated with a fecal microbiota transplant that I will discuss further. The antibiotic treatment of the first infected group lasted for 3 weeks. They put antibiotics in the drinking water to deplete the gut microbiota, and then they were taken off of the antibiotic treatment. And 2 days after being taken off of the antibiotic treatment, they were injected with leptospiral interrogants. The second group was control, so nothing was done to them. And then the third group with the fecal microbiota transplant also underwent antibiotic treatment in their drinking water for 3 weeks. They were also injected 2 days after being taken off the antibiotics, except the only difference is that they were treated with fecal microbiota transplant. What that is that the fecal pellets of the control group were collected, suspended, and spun down to form a solution. And then that solution was given orally for 4 consecutive days after being injected to one of the infected groups. Now, they did that because the gut microbiome of the control group was still normal. It was still plentiful. Whereas if they tried to do a fecal microbiota transplant with an infected group, then there wouldn't be much bacteria that comes from the microbiota to be transplanted. Out of all the groups, they had their fecal pellets analyzed through 16S rRNA sequencing, which showed that bacterial richness and biodiversity of the gut microbiome was not significantly changed, while the relative abundance of firmicutes and bacteroidetes was significantly changed between the infected versus uninfected. So this is the infected group without the transplant. This is directly after being injected. Firmicutes are blue and bacteroidetes are red. This is 2 days after being injected, and this is 7 days. So you can see a significant increase in the amount of firmicutes compared to in day 7 compared to directly after and day 2. This confirms a composition change in the gut microbiota of the mice. Now, the effect of leptospira interrogans on all 3 groups was weight loss. So the infected group without the transplant, 4 days post-infection, had lost significantly more weight than the other 2 groups. The burden of leptospira interrogans also showed an increased leptospiral load in kidneys, liver, and lungs, which will be shown on the next slide. And fecal microbiota transplant confirms that microbiota aids in anti-leptospiral activity. This will also be shown on the next slide. So this is with the control group and the infected without transplant. So 2 days post-infection or post-injection, the infected group had a significantly more amount of leptospiral load in the liver compared to the control group. This is also shown in the lungs. It's not as significant. 4 days post-injection, the leptospiral load in the liver for the infected and the control group decreased significantly, while the kidney and lung leptospiral load increased. So, since the fecal microbiota transplant was done for 4 consecutive days, this is 4 days post-injection, it shows a decrease in the leptospiral load in the liver and lungs. The relative significance of this research was that the composition of the gut microbiota was altered through infection. It also shows a significance or benefit to probiotic therapy through the negative effects of antibiotic treatment. So antibiotic treatment kills good and bad bacteria in the gut, which increases susceptibility to infection. So if introducing more good bacteria, it can potentially fight off infection. These are the locations of research, and these are the authors and my reference. Thank you for your time. Please post questions.